Greetings! Welcome to the lecture of Plant Molecular Biology. In this lecture, I'm going to discuss with you about somaclonal variation in plant tissue culture. I'm Wilson Yong from the Biotechnology Research Institute, University Malaysia Sabah. Somaclonal variation is the term used to describe the variation seen in plants that have been produced by plant tissue culture or regenerated from cultured somatic cells. Plant tissue cultures isolated from single explant can show variation after repeated subculture. Several terminologies like cheliclones and protoclones are referred to the plants which are regenerated from stem callus and leaf protoplast respectively. Similarly, gametoclones are the plants which are regenerated from haploid cells in culture. For distinction, types of variation arise by specific culture of cells or tissues are named after the culture types, such as protoclonal variation in protoplast, gametoclonal variation in anthers and microspores, cheliclonal variation in callus, mericlonal variation in apical meristem, and somaclonal variation in leaf, stem, root, or other somatic tissues. In broader sense, somaclonal variation also encompasses the changes observed at the level of cell cultures that have been utilized to select cell lines tolerant to extreme environmental stresses, resistant to diseases, or for higher productivity of industrial phytochemicals. Its occurrence is both a boon and a bane. On one hand, the somaclonal variation can produce desirable genotypes as novel cell lines or plants of agronomic and commercial advantages. On the other hand, when the objective is clonal multiplication of valuable true to type elite clones, this variation can result in unwanted off-types that reduces the commercial value of the propagules. Similarly, genetically engineered plants are carefully screened to avoid unwanted and unintended somaclonal variation so that the commercially released clones have only beneficial effect from the transgene. Somaclonal variation can be genotypic or phenotypic, which in the latter case can be either genetic or epigenetic in origin. Typical genetic alterations are changes in chromosome numbers, which include polyploidy and aneuploidy, chromosome structure caused by translocations, deletions, insertions and duplications, and DNA sequence resulted from base mutations. Typical epigenetic related events are gene amplification and gene methylation. If no visual morphogenic changes are apparent, other plant screening procedures must be applied to detect somaclonal variation in plant tissue culture. One of the more frequently encountered types of somaclonal variation results from changes in chromosome number, that is aneuploidy, polyploidy, or mixoploidy. Aneuploidy refers to a numerical change in part of the chromosome set, whereas polyploidy refers to a numerical change in the whole set of chromosomes. Aneuploidy is the presence of an abnormal number of chromosomes in a cell that does not have a difference of one or more complete sets of chromosomes, while polyploidy is the condition in which a normally diploid cell or organism acquires one or more additional sets of chromosomes and has 3 or more times the haploid chromosome number. Mixoploidy, on the other hand, is an organism having an unequal number of chromosome sets in adjacent cells or tissues. For example, in the case where 2 cell lines, one diploid and one polyploid, coexist within the same organism. Changes in ploidy originate from abnormalities that occur during mitosis. For example, extra chromosomal duplication during interphase, spindle fusion or lack of spindle formation, and cytoplasmic division. As plant cells grow in age, the frequency of changes in ploidy increases. Therefore, changes in ploidy observed in cultures and regenerated plants might have their origin in the source of tissue explants use. Another cause of variability due to changes in ploidy is the in vitro culture regime itself. The longer the cells remain in culture, the greater is their chromosomal instability. In addition, the composition of the growth medium can trigger changes in ploidy. For example, both kinetin and 2,4-D are implicated in ploidy changes, and cultures grown under nutrient limitations can develop abnormalities. Selecting a suitable explant and an appropriate culture medium can therefore enhance the chromosomal stability of the culture. However, high variations of ploidy in cultures do not always lead to high frequencies of somaclonal variation in regenerated plants. This is because, in mixed cultures, diploid cells appear to be better fitted than aneuploid or polyploid cells for regeneration, as they are more likely to form meristems. Structural changes in nuclear DNA appear to be a major cause of somaclonal variation. The changes can modify large regions of a chromosome and so may affect one or several genes at a time. These modifications include the following gross structural rearrangements: deletions, loss of genes, inversions, alterations in gene order, duplications, duplications of genes, and translocations, segments of chromosomes moving to new locations. Activation of transposable elements can also be a cause of somaclonal variation. Transposable elements, or transposons, are mobile segments of DNA that can insert into coding regions and cause gene disruption. Tissue culture-induced mutations associated with transposable element activities have been reported from many plant studies related to stress conditions during tissue culture, such as wounding, exposure to hormones or specific compounds in the growth media, and genomic shocks from cytological changes. Transposable elements and retrotransposons can function as insertional mutagens in plant genomes, whereas widespread activation can result in a bunch of chromosomal rearrangements, leading to misregulation of genes, aneuploidy, and new transposon insertions. In addition to larger modifications of nuclear DNA sequence, changes at the level of a single DNA nucleotide that occur in a coding region can also lead to somaclonal variation. For example, point mutations that result from a change of base in a single nucleotide or the altered methylation of a base can lead to gene inactivation. Epigenetic changes that cause somaclonal variation can be temporary or transient and over time are reversible. However, sometimes they can persist through the life of the regenerated plant. Epigenetic changes may be caused by DNA methylation, DNA amplification, or by activation of transposable elements or transposons. DNA methylation is important for both transcription and translation because it alters the substrate affinities of enzymes that are active in these processes. Tissue culture media can change the level of DNA methylation and thus may be one of the important causes of somaclonal variation. Over the years, considerable research on trying to identify the causes of variation in culture has been conducted. Apparently, every possible factor that could result in a genetic change has been accounted for as a cause for somaclonal variation. The variation may also occur due to changes in organelles, DNA, enzymes, protein profile, and cytoplasmic genome. Factors that contribute to somaclonal variation are of categories which include physiological, genetic, and biochemical. Variations induced by physiological factors were identified quite earlier. Such variations are those induced habituation to plant growth regulators in culture and culture conditions, and are often epigenetic. They may not be inherited in Mendelian fashion. Prolonged exposure of explanted tissue to powerful auxin, such as phenoxyacetic acid or 2,4-D, often results in variation among the regenerants. Tissue culture regenerants show certain variations which are results of alterations at the chromosomal level. Although the explanted tissue may be phenotypically similar, plants often have tissue comprised of diverse types of cell. In other words, there are cytological variation between the cell types within the explanted tissue, and such a pre-existing condition often results in plants regenerated from the tissue that are dissimilar. These species are referred as polysomatic species. Somaclonal rearrangements such as deletion, duplication, and somatic recombination are the chief sources of genetic variations exhibited by somaclones. In tissue culture, several biochemical variations have been identified in various crop plants, and some of these variants show Mendelian inheritance. But many may be epigenetic and may be lost in the regenerated plant. Biochemical variations include alteration in carbon metabolism, leading to lack of photosynthetic ability, starch biosynthesis and carotenoid pathway, nitrogen metabolism, and antibiotic resistance. While genomic DNA exhibits normal methylation patterns, prolonged exposure of plant tissue to in vitro culture may result in the alteration of normal methylation pattern. Studies concerning different aspects of somaclonal variation are important for several reasons. First, it is hailed as a novel source of genetic variation, which successful utilization depends upon its systematic evaluation and judicious utilization in breeding programs that needs appropriate experimentation. Second, somaclonal variation is of interest as a basic genetic process, since it contradicts the concept of clonal uniformity. It is thought that the cell passes through the stress, as a result of which the genome, known for its plasticity, restructures itself to modulate the expressions of genes as demanded by in vitro conditions. Third, somaclonal variation is unwanted when the objective is micropropagation of elite genotypes or genetic transformation which partly involve tissue culture. It needs minimization of variation, which may be achieved through manipulation of media components, explant source, culture conditions, and etc. Mutagenic agents, both chemical and physical, such as ultraviolet or UV radiation, ethylmethane sulfonate or EMS, nitrosoguanidine, and sodium azide, are used to produce various heritable forms of somaclonal variation. UV radiation induces dimer formation between adjacent thymine residues in DNA. This produces lesions that cause frameshift mutations and base pair substitutions during DNA replication. EMS and nitrosoguanidine are alkylating agents that cross-link and sever DNA molecules, often resulting in gross DNA alterations. In contrast, sodium azide induces single base pair substitutions or deletions, resulting mostly in small point mutations. There are several different approaches to detect and isolate somaclonal variants from cultured plant cell populations. Morphologically distinct cells, such as non-photosynthetic or non-green cells, and cells that accumulate anthocyanins or other plant pigments, are detected visually. To isolate herbicide and antibiotic-resistant variants, plant cells are simply grown on herbicide or antibiotic-supplemented media. This method can eliminate wild-type cells and allow herbicide or antibiotic-resistant variants to survive. Direct selection technique can also be used to isolate temperature-tolerant variants, because those cells which survive in an extended incubate period at abnormally high or low temperatures are temperature tolerant. Those somaclonal variants that cannot be detected visually or selected directly are isolated by indirect means, such as for the auxotrophic plant cells or cells that are unable to synthesize a particular organic compound required for its growth. When grown in the absence of the necessary nutrient supplement, these cells become very weak, and at this weakened state, these cells assimilate exogenously supplied cytotoxic compounds such as arsenate, bromodioxyuracil, or fluorodeoxyuridine at lower rate than that of wild-type cells. Thus, treating a cell culture under restrictive growth conditions with one of these substances would tend to favor short-term survival of the weaker auxotrophic cells, but these restrictive conditions will not affect the normal wild-type cells. In conclusion, several strategies have been used to determine the genetic fidelity of the in vitro produced progenies, in view of the fact that the commercial viability of micropropagation technology is reliant upon maintenance of genetic fidelity in the regenerated plants. Tissue culture-induced variations, while posing a significant threat to the genomic integrity of regenerated plants, also provide plant breeders with tools for improvement, especially for crops with a narrow genetic base. With that, I would like to bring my presentation to an end. I hope you find the lecture informative. Thank you very much for attending and participating in this lecture. Thank you.